Good morning, everyone, and welcome to our monthly live session of peripheral intervention cases from Mount Sinai Hospital, New York City. Today, good morning, March 24th, 8:05 Eastern Standard Time. I welcome to you for another case right here. Before we move into the lab, just want to introduce a special friend of mine. I'm sure everybody in the coronary world knows Dr. Usman Babar. He's here to give some insight on now in the peripheral world, so he's probably moving to the dark side. But before we do that, just wanted to give you run-of-the-mill some homework. Our cases go in the archive section on ccclivecases.org. So any of your previous cases from previous months are in the archive section, and this case will go by the end of the week for any of you to view on them. Comments, suggestions, concerns, questions, please do email us on the link. And also, just to give you a heads up, we have our annual Link Mount Sinai conference coming up this summer between June 10th and June 12th. June 10th will be the endovascular fellows course concentrated session, and 11th and 12th will be more of an endovascular approach with all the great talkers and presenters from all around the world, as you know, associated with the peripheral world, will be here. The agenda will be up on the website pretty much soon, by the end of the week or in a week or so. But we would love to have you there. Please do come, participate, join us, discuss, and enjoy this annual event of ours. So without further ado, morning, PK.
Good morning, guys. How are you? So first of all, it's a real thrill to have Dr. Babar here, who's going to really help us understand some of the things that we've been struggling with in the peripheral world with the new DCB data that's come out. But we first have a really interesting case, which again, like what we try to do here is show you what to do and also what not to do. And part of it is a workup of a patient who has severe claudication. But let me first get to the case, and then we'll go ahead and discuss with Dr. Usman about what he's here to help us with. But let me introduce the team here. We have to my right Dr. Karthik Guja, our Associate Director of Endovascular Services here at the Mount Sinai Hospital. Next to him, Asma Khalik. Asma is our endovascular fellow, who all of you know from multiple different cases that we've done live. Next to her, we have Sardar Farhan, who's here from, who's also our second endovascular fellow, who's spending another year with us. It's been great to have him with us. Behind us, we have Ray Lascano, we have Elizabeth, and we have Damien as well. And so we're ready to go. We have a great case here with a lot of issues that we didn't expect, so I guess that's part of the fun of doing live cases. So without further ado, I'm going to ask them to go ahead and present the case, whoever's mic'd up, to go ahead and start.
Good morning, everybody. So we have a 70-year-old female with a past medical history of hypertension, hyperlipidemia, diabetes, hypothyroidism, former smoker, known CAD, multiple PCIs, known PAD with status post aortobipham bypass, as well as prior carotid stenosis, status post left carotid endarterectomy, who essentially came in as a claudicant, complaining of severe bilateral lifestyle limiting claudication, limiting her to 2 blocks of and it resolves with rest. She is on regular medications, aspirin, Plavix, and diabetes medications, normal labs. We did some basic workup on her. Her ABI PVRs are reduced on both sides, 0.87 and 0.86 on left and 0.87 on right. Vascular ultrasound showed that she has severe plaque on the right with moderate to high-grade stenosis at multiple locations, and left also shows severe plaque with areas of moderate to high-grade stenosis at multiple locations in the SFA, both of these. Now, next, please. So this is a runoff from a previous day when we went from the left side, went up and over. You can see significant disease mid to distal SFA. And so essentially, the next picture will show you. So go to next, please. And the below the knee runoff was 3 vessel. Now this is a still frame from an iliac angiogram, and it's the aortobipham, and you can see how tight the iliac bifurcation is, the bifurcation of the two, the aortobipham is. So essentially precluding the contralateral femoral axis, which is how we had done the diagnostic anyways. So then we plan to bring her back for an alternate access to treat her left side, because her left side has worse symptoms. So today we were all yeah.
So at this stage, let me. So part of the workup, I think, what everybody should know when we're planning on getting alternate access is obviously to make sure that certain things are done, prior to doing it. So so this young lady is obviously a vasculopath. Let's check an ACT, guys. Is a vasculopath, and she has an aorta bifem. And I guess one of the things that was missed was whether or not she has subclavian artery stenosis. So if you're planning on going from the radial, you need to have at least a patent subclavian to try to be able to go. And like I said, part of what we try to teach you is through our mistakes, you don't make these kind of errors in terms of workup and preparation of the patient. So you can see here that we were a little disappointed and surprised when we saw the following. And I'm going to go back here. I'm going to show you. So this is what you want to do when you do any sort of radial access is standard radial access with a woolly wire. I know the cardiologists, which I am a cardiologist, love to use a glide wire. But the truth of the matter is, the glide wire really could be a very dangerous wire in the wrong hand. So we tend to go with a woolly wire, which allows us to be able to track up without a problem. As you can see, it crossed this heavily calcified subclavian without a problem. And even if it had got into any of the branches, there wouldn't be an issue. So once we crossed, we had trouble. We took a picture, and I want Dr. Baber to react to this. And you can see that we're dealing with a very, very tight subclavian. And you can imagine if we're planning on doing a radial intervention through this is going to be a real issue. So at this stage, so what we did was we went ahead and we with some difficulty, we were able to switch out to an IMA catheter, torque the IMA catheter to point down, and then went ahead and put the wire all the way down into the distal aorta at the level of the aorta bifem bypass. The nice thing, did we give an ACT right? We gave an ACT? Good. The nice thing is Terumo and other companies have started to make radial sheets that are eligible for below knee intervention or below inguinal intervention. Definitely you can do iliacs from the arm. We've done that for years, and renals from the arm. But this is the kind of case where this young lady is probably going to end up getting an antegrade if we're not able to get through this. So Terumo makes the 5/6 slenders. I believe they make them in what do they make them? 135 and
we have
and one at a time. I'm sorry.
120 and 150.
Okay, so they make them
We
in
have a
120 and 150, and they have balloons that are in the shaft lengths of 200. And I believe as this entire sort of system starts, what's going to happen is you're going to start to get slow. Please, is she jerking the subclavian? The rail. You when the wire moves, you're not railing enough because the wire can't move. So you're gonna start to have different kind of systems for you to do atherectomy and other things from the arm. So at this stage, we've given 5,000 of heparin. She's quite comfortable. Our plan is to use an 014 balloon to dilate this. And then if we can, it doesn't look like it's going to happen. We're going to have to go antegrade here. So it looks like what we're going to end up doing is using an 014 balloon to dilate if we can. If we can't, then we'll just go ahead and do antegrade stick and take care of the case. So, one of the things that you always want to watch out for, as I always tell everyone, is you have to know your bailouts. It's not going to go. You have to know your bailouts. And your bailouts have to be an issue. So in this case, we have a 5/6 slender in. We have a 7029 VBX ready in case we need to stent after we balloon. The problem is you're going to be working on an 014 wire, which should be okay. So we're going to go ahead and put an 014, 018 wire. It doesn't really matter. And then we're going to go ahead and balloon. So in the meantime, since Dr. Baber's here, I'm going to go ahead and ask Dr. Asma to go ahead and really talk to Dr. Baber and all of us about this meta-analysis that's really created so much news. As all of you know, and Dr. Guja and myself and Dr. Kapoor have implemented this in the cath lab, where currently we are not using drug-coated balloons because of the new FDA letter that recently came out. So I asked Asma if she could kindly just kind of go over this meta-analysis, and then obviously Dr. Baber, who's on the editorial. Actually, Usman, forgive me if I'm wrong in your title. I know that you have a very high position in JACC electrophysiology in the editorial section, and especially in statistics. So I know that no one better than you, especially to explain to all of us on what the take homes of this kind of study should be, what are the strengths and the flaws of such a study, and so on and so forth. Anything else that you might be able to add?
Okay.
So while I'm holding this sheet and we're working online, I'm going to have Asma go ahead and present, and I welcome any comments from Usman or from obviously Dr. Kapoor to go ahead and about the situation we're in.
Great, thanks, BK. It's Usman here. I think my title is Super Fellow, actually, but no, I think I'm beyond that. So happy to be here, and looks like you guys have caught yourself in an unanticipated situation. So actually excited to see how you're going to get out of this. But yes, if Asma wants to kind of give a brief, maybe a brief
Can we get the
summation, and
slides?
then we can kind of talk as you're dealing with this subclavian issue. I think that'd be great.
Great, thanks, Usman.
Thank you. So could we get the slides, please? Okay. So well, we can talk about the peripheral radial equipment in a second. So next, please.
729 goes on to 7 French.
Next.
Yeah. So the next, please. Yeah. So basically, this is the recent study that created a lot of controversy and stir up in the drug-coated technology and use in peripheral interventions. So it's Dr. Katsonis's meta-analysis that was published in JAHA in the last year. So essentially, what they looked at is it's a meta-analysis of several randomized trials using drug-coated technology, both balloons and stents, and essentially looking at 28 randomized controlled trials and almost 5,000, 4,600 patients, and they looked at the 1 year, 2 year, and 5 year all-cause death in all these studies. Next. So a little bit background of how they picked up these studies was they essentially had to be randomized studies, and they chose out of over 300 studies. Eventually, their criterion they only were met by 28 studies, and which I already said had around 4,600 patients. Next, yeah. So they did this very fancy statistical analysis, which I would let Dr. Barber explain to you guys later. And they derived at a few conclusions, which I'm going to touch upon here before handing it over to Dr. Barber. So essentially, their conclusion was that at 1 year, they looked at 1 year all-cause mortality, and at 1 year they did not see any good evidence of pooled risk of death in these studies in studies that used in the patients who got paclitaxel coated drug or stent therapy versus control arm. Next. So essentially, no significant effect. No significant effect was noted at 1 year between the two. Off-crew death rate and the relative risk of death was just 1.08, so which is non-significant. Next, just to note that most of these patients were claudican. About 90% of these were claudicants. And at 2 years, about 12 studies had a follow-up of up to 2 years out of these 28 studies, and that included around 2,300 patients. And they concluded that at 2 years, there's good evidence that the application of paclitaxel-coated devices in the fem-pop arteries was related to significantly increased risk of death. So they showed that there was a 7.2% crude risk of death in the paclitaxel arm versus 3.8% crude risk of death in the control arm, in the non-paclitaxel arm. And they calculated a relative risk of 1.68, which was statistically significant. And they showed an absolute difference of 3.5% and number needed to harm, so meaning that 1 out of 29 patients was going to die from using this therapy. Next. This is the same thing put in a forest plot, essentially the same thing which I just mentioned. And here there were about, like I said, 2,300 patients or so. Next. Now, looking at 5 years, they showed that the long-term analysis of all-cause death of up to about 5 years, there were only 3 studies that showed that much follow-up, and they included around 863 patients. And out of these, they showed that 78 deaths out of the 529 in paclitaxel arm, meaning a crude risk of death of 14.7%, versus 27 deaths out of the 334 patients in a control arm, giving it a crude risk of 8.1%. So showing that the pooled risk ratio was 1.93 and the absolute difference was 7.2. And again, they showed at 5 years the number needed to harm was even lesser, surprisingly just 1 in 14. Next. This is the same thing shown in a forest plot. Like I said, only 3 studies were used: Thunder, Impact, and Silver PTX. They had a 5-year follow-up. Next. So then I just want to bring up one more point. Then they did this another fancy statistical method called the meta-regression, and they were trying to. So I'm directly quoting it from the paper, saying what they said was that to establish the epidemiological evidence for causation, they try to explore the presence of biological gradient. That is, whether the greater dose exposure to paclitaxel increases the risk of death. And what they found out was essentially that it did. Next. So they created this very fancy formula of studying this exposure with the paclitaxel dose time product, they called it, and they essentially next. Yeah, so what they found was that the random effects meta-regression, it identified that there was a significant association between the paclitaxel dose time product and the absolute risk of death. So in the graph here, you can in the diagram here, you can see that. So the risk of death in the those subgroups that were 3.5 micrograms per millimeter square of paclitaxel balloons, they had a relative risk of 2.3, whereas the ones that eluted 2 micrograms per millimeter square, they had a non-significant risk of death of 1.27. So it is essentially, at least according to them, they're hypothesizing that it's a dose-related effect, and they also were able to calculate that any 0.4 plus minus 1%, that there's a 0.4 plus minus 1% excess risk of death per paclitaxel milligram year, with the p-value of 0.001. Next. So then this is from their talk presented at from the same group. It was presented at Link, and I just borrowed a couple of slides from them. They demonstrated in this slide that the TLR at 2 years was again a dose effect, which we all know. The more the balloons or stents, the more they elute the drug, the better they are at preventing TLR. They categorize them into 2 groups of using less than 3.2 milligrams versus using more than 3.5 milligrams. And it showed that that the subgroups that were using more than 3.5 milligrams essentially had they were better at preventing those devices were better at preventing TLR. And go to the next slide, please. Yeah, but the similar effect was seen with death as well, meaning that the subgroup of drug-eluting therapy that had less than 3.5 milligrams was not, in the random effects model, was not significant. The relative risk was non-significant for death, versus the subgroups that were eluting more than, or that had more than 3.5 milligrams of drug, they were more likely to cause death. Next. So just a quick overview of what these deaths were. They were predominantly cardiovascular deaths, and with a relative risk of 1.73. Some of them were infection-related deaths as well, again with a relative risk of 1.75, and respiratory adverse events and gastrointestinal adverse events. And this is, seen across all of the balloon types and the Zilver PTX. Next. So now their conclusion was that, like I said, before the risk of death was identical at 1 year, and at between 1 to 2 years, the mortality is increased. And again, there's a significant relationship between the dose time exposure to paclitaxel and the incidence of death. And again, the exposure formula, they derived at this number of 0.4 plus minus 1% per paclitaxel milligram year. Next. So this led FDA to produce an updated, warning letter, the usual Dear Physicians, Cardiologist letter that they send out, and basically saying that the treatment of peripheral arterial disease with paclitaxel-coated balloons and stents is potentially associated with increased mortality, and it was a letter to the healthcare providers. Next. In which they said that that we have now concluded that they did their own preliminary analysis, their own independent preliminary analysis of the long-term follow-up of some of the studies of the pivotal pre-market randomized trials for paclitaxel-coated products, which essentially their analysis was kind of in line with what the original meta-analysis was. And they said of these 3 trials with a 5-year follow-up, each showed high risk of mortality in the subjects who were treated with paclitaxel-coated therapy. And essentially they said that in their analysis, 975 subjects in the 3 trials, and they said that approximately 50% increased risk of mortality in the subjects who were treated with paclitaxel-coated therapy versus control. And again, their crude risk was even more magnified, 20% versus 13% risk of crude risk of death at 5 years. Next. And then they came out with these recommendations that there's a continued and diligent monitoring of these patients who've been treated with these therapies. And again, while they're going to, for their final recommendations, they're going to wait for more prospective and more long-term data, but they obviously gave a word of caution, and that they recommended discussing before subjecting patients to these treatment options, discussing with the patient the long-term or potentially conceived long-term effects of this kind of drug therapy. And again, they said that some individuals, particularly who are at high restenosis risk, in them the risk may outweigh the benefit may outweigh the risk of therapy. And again, the usual lifestyle modification, etc., which we already do, they should be promoted. That's all I have. Thank you. UB?
We can do a stent here. Okay, I'm sorry guys, we're working on this. Let me show you a little bit about what we've been doing, guys.
Where do we stand?
So what we did in the meantime, while Oscar was doing that great presentation, we went ahead and ballooned with a 5. Then once we ballooned with a 5, we tried to track the sheath up and over, but it wasn't going, so she was having a lot of pain in the arm. So what we may do is stent the subclavian from the arm today, and then bring her back for the leg, and take care of that from the leg. So do antegrade access or go through that stented subclavian. But in the meantime, we have a good pressure. But what's happening, and I want to show everybody, is that when you have resistance to push, what's happening is the vessel in the arm, the radial in the arm is bowing,
Spasm.
and she's having tremendous pain in the arm. So I think at this stage, what Dr. Guja and I feel is we're probably going to do balloon angioplasty and stent of this subclavian artery from above, and we can demonstrate that from the arm. And then we'll go ahead and either bring her back, go through that stent to go ahead and do the distal SFA, or we'll bring her back and do antegrade. But as he decides on what he wants to do with this, I just want to go ahead and ask Usman. Usman, you saw this presentation. Yeah. We've gone over it, Vishal. We all sat together and talked about it.
Right.
This has created really the ultimate blow in the sense for all of us who work in the endovascular field. Usman, for us, we were fraught, as you know, with claudication. These patients aren't coming back, aren't going to die because of their claudication symptoms. We want to make them feel better. And, with all the different technologies that were available, including stents, including atherectomy, including covered stents, we had this huge problem of restenosis, similar to what the coronaries have had many years ago, back when I was training. So, the DCB really made a difference. I mean, in our practice, myself, Dr. Guja, Dr. Kapoor, we were seeing a lot less restenosis come back from using this drug-coated balloon. So it was sort of like a boon for us. So obviously with this and the new FDA letter, I don't know if Asma went over it, I was concentrating over there, and the FDA recommendations, at Sinai, we have stopped. I mean, not only us, but our radiology colleagues, our vascular surgery colleagues have all felt that this is not something that we should be doing. So I just wanted you to break it down because this has become a real point of contention and a controversy for us. And I have 3 questions for you. One is can we take this as the Bible from this meta-analysis? I mean, is this something that we can interpret and say yes? And second is, what is it that we need to prove this wrong? If this is for instance what a correct statement that this meta-analysis is making. So I'll let you break it down. Go ahead.
No, thanks so much, PK. It's glad to be here and glad to make some brief comments, and thanks for Asma for kind of going through the sort of the summation of the lay of the land right now. And I totally agree with you. I think the field is obviously confused. There's many rapid evolutions. We see different papers coming out all the time. And trying to distill this with the rapid pace of evidence generation can certainly be challenging. And I totally agree with the decision, I think that you made and that we made as a hospital to sort of be cautious and to hold off on implanting these devices until we get a little bit more certainty in the data. And I think there are sort of 4 questions that have emerged, none of which are answered, by the way. First question, is there an actual signal of mortality with the use of paclitaxel-coated devices? And if there is, what's the magnitude of that signal? Is it related to just balloons, or is it also related to stents, or is there a differential effect? Is there a time dependence? In other words, is the risk only late and not early? And then finally, is there sort of a dose relationship? And obviously, all of this started with this meta-analysis that we just heard about. And with respect to saying, should we take the meta-analysis as sort of the Bible and just put our hats on it, my short answer is no, because meta-analysis doesn't provide us that level of evidence. Nevertheless, there are some interesting findings, and I think it's important to understand some of the strengths and weaknesses. Meta-analyses are good when you're trying to identify sort of signals that are hard to detect. These were small studies, not powered for mortality, and you can get power by trying to pool information together. This is what happened with stent thrombosis in the coronaries, by the way. None of the coronary trials were powered for stent thrombosis. It wasn't until large meta-analyses were done where we began to understand the risk with new generation, early generation stents. So I don't think it's wrong to take an approach of a meta-analysis, but let's understand the strengths and weaknesses. The most important thing, I think, is that the average follow-up in these meta-analyses was about 2 years.
Right.
And whenever you have time to event analyses, when you get beyond your average follow-up duration, your power and precision drops off. That's just a simple statistical fact. And so this finding of this magnitude of doubling of risk at 5 years, I think you have to be a little bit cautious about that. There's very few events, only 3 trials went that far, and that's well beyond the median duration of follow-up for all the trials in aggregate. So that's just something to be aware of whenever you're doing statistical analyses, is your power and precision to detect differences drops off beyond your sort of median follow-up time. The second point, and this is, I think, becoming more apparent, I wasn't even aware of this when we first discussed this, PK, has to do with really the quality of the individual trials. And as I mentioned, none of these trials were powered or designed for mortality. It wasn't part of the primary or secondary endpoint. And if you look at the trials, the 3 trials that have 5-year data, Thunder, Zilver PTX, and Impact SFA, 2 of those trials, Thunder and Zilver PTX, had follow-up, loss to follow-up or withdrawal rates in excess of 30%. Now that has a very important implication, because that means when those patients are lost to follow-up or withdraw consent, you don't know what happened to them afterwards. And as far as I know, and all the reading that I saw, I don't think anyone knows what happens to these patients, and I don't know if the FDA is looking into that right now. But unless you know what happened to that population of patients, are they sicker? Did more of those patients die in one arm versus the other? I think it's very hard to make definitive inference on whether or not there's a higher mortality in one group or the other. So, and the final issue is this dose response thing with the meta-regression. I think meta-regression, you have to be very careful with how you interpret that. Remember, meta-regression is trying to make analyses at study level. We want to know what's happening at the patient level. And in fact, the IMPACT SFA patient level analysis published in JACC showed no association, no dose response effect in contrast with the meta-regression. Having said all of that, I think one has to accept that there seems to be some signal. The FDA has put out their letter. The meta-analysis suggested this. What we don't know is what's the actual magnitude of that signal. The meta-analysis suggested a twofold risk. The FDA has said it's about 50%. The truth is probably, I'm not going to say what it is, but there's probably some signal. The question is, what's the magnitude of that signal? And that still needs to be sorted out. And then we need to do some more in-depth analyses to figure out, is this uniform balloon or stent? Is it time-dependent? So on and so forth. I think the best way to do this is probably 2 exercises. Exercise 1 would be to get the patient-level data of every single long-term study and particularly look at the vital status of patients who were withdrawn or lost. I think that's crucial. You've got 30% or so patients who are withdrawn or lost. You don't know what happened to them. So until you know that, you cannot be certain about the magnitude of effect and whether or not there's an effect. So I think that has to be the very first thing. I'm sure that's what the FDA is doing. That's what I'm going to assume that they're doing. They're taking all these data, putting it together, figuring out all the patients who were lost, and they're going to convene their circulatory or device panel and probably put out something more definitive. That, I think, is the bare minimum. And I think it would be interesting to see what happens after that. The best way to answer this question is to do a trial. The best way to answer this question is to do a trial powered for mortality. And when you have something of this magnitude of public health consequence, as you just mentioned, it's changed practice. I think if the public health impact and if the patient level analysis by the FDA is suggesting, I would be surprised if they don't mandate an actual trial be conducted powered for mortality. I mean, this is what happened with dual antiplatelet therapy. This was a big question. No one knew what to do. The FDA, in concert with academics and industry and other stakeholders, put together the DAP trial. And we got an answer.
Right.
And it was a public health question. So I think when you get into issues of public health and mortality, you need a definite answer. We don't have that yet. We have suggestions. And I think that might be the best way to answer the question.
So Usman, I mean, that's a great summary, and it really helps me understand what the value of this meta-analysis is. But one of the things that bothers me is that I was the national PI of one of the trials, and and there are different balloons and there are different efficacy rates across. If you look at all the different technologies that are available with paclitaxel, so if you look at the stents, they have a different efficacy. I know there's an issue with one of the stents and how the trial how it was reported, but the question was that if they're not all equal, other than the one common denominator is paclitaxel, not even the dose, because if you look at the doses of paclitaxel in each one of the balloons, it's different. If you look at the excipient or the mode of transmission, for lack of a better word, of the paclitaxel, it's different as well. So my question to you is, how can you generically say that paclitaxel in PAD causes mortality when the dose of paclitaxel is so low as compared to what they use in breast cancer? Paclitaxel had been used for years in the coronaries, as you know very well, without this type of signal. We know that you had stent thrombosis, but it wasn't a mortality, just a generic mortality signal. So how can we, those of us who have used it, take that and, how do we interpret that? Could it just be a statistical anomaly that this happened? Or is this something that we need to be concerned about? Because we have not seen it in any of the trials. If you look at the IMPACT trial, like you just talked about, every death was adjudicated. Yeah. And if you look at if you look at the pharmacokinetics of paclitaxel and the time to death, the paclitaxel was out of the system. Yeah. So this is why I get frustrated as an endovascular interventionalist, because I know this stuff works. I know that we're not having we're lowering restenosis rates, and now this great technology is taken out of our hands because of what may be just a statistical anomaly.
Yeah. No, I mean I completely hear you, and I think the short answer is I don't think you can definitively say we know for a fact that you can make causal inference that the implantation of paclitaxel devices increases death. I don't think a meta-analysis can give you a causal inference.
Right.
There is statistical heterogeneity and there's clinical heterogeneity. The meta-analysis didn't find statistical heterogeneity, but you just pointed out obviously there are differences in the devices and the patients, the type of trials, the methodology. So certainly there's some degree of clinical heterogeneity. So a meta-analysis should not be the level of evidence for which one makes a causal inference. So the question becomes is why are we seeing this signal? And that's what I'm saying is we don't have an answer to that quite yet. Is it device dependent, as you're perhaps suggesting? Is it related to a dose response relationship? That's also a possibility. And the third point is it just noise?
Right.
Is it just a false positive result? And I think that is also has to be taken into consideration given the degree of follow-up in some of the studies, particularly in the 5-year data. So we don't have an answer yet. We don't know if this is a true finding or sort of a spurious finding. I do not think a meta-analysis gives you the level of evidence to say that definitively, which is why I think the FDA did not say, explicitly say do not use these devices. They had a more cautious interpretation. Because to sort out the issues that you're saying, is this an issue related to one device or another? Is this an issue related to some kind of dose? Is this an issue related to some time of follow-up? The only way you're going to know that is do rigorous patient-level analyses with all the available follow-up data. So you've got to find all these patients who were lost, figure out their vital status, and properly analyze the data to start seeing the proper trends. I believe even when you do that, you're not going to have probably the numbers to make, again, definitive assessments. Again, Zilver PTX is completely different than, Impact SFA, that is completely different than Thunder. And neither one of these studies are individually powered for, the endpoint of mortality. So my suspicion, maybe I'll be completely wrong, my suspicion is that the FDA will convene, they'll put these data together, there will still be some concerning signal. The only way you're going to answer definitively, I think, for the community is probably do a definitive trial.
So the other question I have to ask you, Usman, is what happened with Silver PTX? I mean, we all read our journals are all incredibly respected and all this stuff. You know that there was a flip-flop of the mortality data. With how does that happen? I mean, I honestly, I mean, I don't mean to be in any way disrespectful to anyone. I'm just, I'm curious because I mean, you're obviously on the editorial board of multiple journals and you're part of a, you're extensive reviewer of so many articles, and I know that there's so many checks and balances, and and if you saw. In the I believe in the table it was correct.
Yeah, the
right.
table
In the
was
table
correct.
it was
the
correct.
Abstract part was
and in the abstract it was wrong. So it was sort of like well, what happened? That kind of thing is what I'm kind of asking. Is that even a possibility for that to happen?
And the same thing. In Impact SFA, I was just reading that this morning before coming here. Impact SFA, you had numerical reduction in the original patient meta-analysis in JACC. And then the authors came back, and actually the hazard ratio that Impact SFA is reporting now is a 1.7 for mortality against the paclitaxel-coated device. Granted, that's lower than the 1.9 reported in the meta-analysis, but certainly a concerning finding. As far as how I think it's just really, really bad luck. There are lots of checks and balances, when you're, and this goes back to the level of the statisticians and the coding and how they write their programs. And, it's quite surprising that this would happen because this is probably checked and double checked and triple checked. Now, when that information gets sent to a journal, and I think this brings up a whole other question because there's a lot of debate now. So if I'm a company or I'm an investigator, obviously I have a bias towards making sure my data look good. I analyze the data. I tell the journal and the community, academic community, the regulatory community that, look, these are the data, here it is. What doesn't happen right now, at least in the most journals, is the journals don't ask for the raw data. And that's become an open question as to whether or not raw data should be sent. Should the data be shared? Should you have an independent verification of analyses? And I think those questions are ongoing and evolving, and there's a big push perhaps for some of these trials for the data to be publicly available so anyone can look and confirm or refute these analyses, and that would take care of some of this. But right now, in the peer review and process in most of your papers, the sponsor analyzes the data, or an independent group analyzes the data, and the summary is sent to the journal and they report it out. So the journal wouldn't know unless they had access to the raw data files, which right now is not routine.
Well, from a perspective of you being so involved in all this, the question is it possible? Is there physical manpower available for journal? Because you imagine the amount of articles that's submitted to a journal. I mean, you're now asking for the raw data and the evaluation of all the raw data. Obviously, academic integrity, research integrity, what we take as a Hippocratic oath for all of us, is part of that integrity thing that we talk about. Do no harm. And by reporting false data, obviously you're doing harm. So I'm not saying that's happening. I'm saying it was an anomaly. It was missed.
Right.
That the fact that the right data was in the table tabular form, and for whatever reason during the writing of the journal, this particular thing was missed. But it was also missed in the check and balance on just plain reading of the article as it was being submitted or accepted. Right. So that's one thing. But my second question or third question to you is, if you look at the 5-year follow-up and the mortality associated, because they report an increased mortality at different times, as you know, in the meta-analysis. But there's a lot of chatter that, well, a lot of the DCBs and drug-coated balloons, for instance, the Imperial studies only reported 1-year data. Stellarex is, the Levant study hasn't publicly reported their 5-year data yet, other than in their registries. So, Stellarex, I know we're in the process of writing and publishing, and we sent out a letter, the Philips company sent out a letter just recently about the fact that at 3 years, they don't see a mortality signal. So do you think at some point it's going to come down to, hey, the 5-year signal that we're seeing really has to do with a couple of products, and I use all of them, so I'm not playing favorites on any particular one here? Or do you think this is going to be a class effect where the FDA is going to say, hey, we need to do a full new trial or whatever analysis on all paclitaxel coated products to make sure there's no safety issue or mortality issue? Because if you look at the quote unquote low dose DCBs like Stellarex and now the Ranger DCB and the Imperial stent, which is, what is the name of that stent again? Illuvia. The Illuvia stent. You could see that they have a lot lower dose than the Impact and the Silver PTX stent. So that's my question to you. Do you think that the FDA will call this a full effect, or do you think it'll just remain as a device-specific effect that they're going to look at?
Yeah. So I think the only way to answer that question really is you have to do sort of what are called interaction analyses, and you would have to see if number one, is there a mortality effect? Number two, does that mortality effect vary by device? I think the challenge is going to be that there's not enough information, not enough studies with 5-year data, not enough patients, so it might be very difficult to definitively say if one device versus another is sort of completely safe. Hopefully they'll have enough power to examine this from a dose response, the amount of paclitaxel that's exposed to the individual, and make some decisions that way. So I think there's more chance that they'll be able to examine dosing in a much more quantitative and a precise manner. But as far as the products themselves, I think that'll be pretty tough, because we're not talking about 10,000 patients. We're talking about just a very small number of patients, even with these three trials. So trying to sort of separate out the effects of one trial versus another, I think it will be almost impossible, personally.
So let me ask you one question, Usman. So from a statistical standpoint, do global registries and follow-ups play any role? I know trials are like wearing a standard, like a custom-made suit, But real-world global registries, would they not give us a better perspective of how really it works in real life?
That's a great point, and I think it depends on the question. So if the question is about efficacy of a therapy, I don't think registries are good because the trials give you the information on efficacy. For safety, I do think so. I do think registries can be very good, and they've been used in that manner to examine. A lot of this was done with early data with stent thrombosis, if you remember when the bare metal stent and DES issue came up, and registries were actually used in very large volume. So I think looking at a very large registry for safety signals is done, and it is useful, assuming one can adequately control for baseline imbalances, and that's the big issue. Registries, if there's a lot of differences in the types of patients who get DCBs or DES versus others, then you're really challenged. But the good aspect of registry, you have a lot of patients, it's real world evidence, and for safety, I think we do look at registries because it tells us what's happening in the real world.
Yeah, because IMPACT has a very good global registry follow-up, and then I guess that could be probably one of the other factors that could, again, give us an indication that is everything pointing in the one direction, or do we have some anomalies? And we already know that PAD patients anyways have a higher risk of CAD.
Yeah.
So is it like just a coincidental finding, or is it truly related to paclitaxel like PK talked about? So
yeah.
should be interesting to see how things go. Anyways.
Absolutely.
So Usman, just for the audience, I know this is incredible. Your insight is I really thank you for doing this. You can see so what we've done is, I just want to show you on subclavian stenting, because we're kind of getting off track in terms of this case now. Rather than fix the leg, we're going to fix the subclavian. So I want everybody at home, I know the radial revolution is occurring, and I get it and all that stuff, but when you're stenting the aorta, the important thing to understand is you need to be able to stent the ostium, as any interventionalist knows. The plaque from the aorta grows into the subclavian, causing obviously the stenosis. So it's very important to hit the ostium and hang into the aorta. Sort of like if you're doing LAD diagonal bifurcation lesion, if you're a cardiologist, this is something that we always teach, that you have to kind of involve the parent vessel in order to be able to truly hit the ostium. But it becomes even more paramount here. So, we in live cases in the conferences, we see it all the time. People go from the subclavian and just go ahead and directly stent the aorta. Stent the subclavian from the arm. But the right way of doing it is from the groin. But in this case, because of the aorta bifem, we did not want to go from the groin with a large sheath. So we used our Terumo sheath that we have in the arm. It's a 5/6 slender. And we're going to go ahead now, but we had to put a 4 French pigtail in the leg. And you can see by the picture that Dr. Guja has done, you've got a pigtail, in the 4 French pigtail in the aorta, and a 6 French slender up in the arm. And now he's going to go ahead and balloon angioplasty with a 6-0 and then use a covered stent because of the eccentric calcium. So I misspoke before. It is actually the life stream that goes through a 5/6 slender. And you know that the 7.0 live stream can go, I believe, up to a 9 or a 10 if we need to post-dilate. So we're kind of ready. But since we had trouble pushing the sheet through, we're going to go ahead now and stent this, and then either offline or bring it back. You can see that the balloon barely goes through as well. You can see how much difficulty this is going to be if we had tried to just stent. So while I'm going to have them put this up in both ends, and we're going to continue the discussion with Dr. Babra as Dr. Guja goes ahead and tries to figure out, and with Ray, on how to do this. So yeah, you can see the balloon just slipped through, and you have to give some positive pressure. So Usman, when you can't explain a causal reason, right, for this, and when you have paclitaxel used in other areas, as you and I both know, but being doctors, why do you have to mandate a trial? Is my point. You're using it in other areas in much higher doses.
Yeah.
And this is in the leg. I can understand if you were working in the aorta, you're working the pulmonary circulation, whatever it may be, right? I can understand somewhere where this is mostly in the legs. We know there was an increased amputation rate in Impact Deep. We talked about that in our fellowship program. But this mortality without a cause. It worries me that we're now going to have to do another trial. As you know, this is going to take a long time.
No, I know, and I think if you go back, to the early mid 2000s, and first generation drug-eluting stents came out, And if you look at the original trials of Taxus and all those original trials, the data were pristine. Very low restenosis rates, no signal for excess harm with stent thrombosis in those studies. And it wasn't until it got deployed into routine practice and there was these signals for excess stent thrombosis well beyond the time point when paclitaxel should actually have been, gone. The polymer degraded and everything, and that led to a change in clinical trial design. That led to public health statements and a real change in practice. So, I think as a scientific kind of individual, I completely agree. It's hard to understand sort of the mechanism here, and I'm not going to pretend that I do. But there's precedence for finding these kind of signals from study data and then trying to investigate exactly whether or not this is real or not. And I think the question becomes, even if you don't have an understanding of the mechanism, the question becomes, do we believe this signal or not, and is this something that's important for public health and clinical practice? And if the answer to those questions are yes, then I think you got to have a definitive answer. A lot of times epidemiology or studies give us signals that we don't quite understand. The mechanism doesn't come until later. I mean, LDL cholesterol we knew for years, was associated, with excess risk for heart disease. We didn't know until years later about the LDL receptor and targeting that and understanding the actual causal pathway. Same thing with inflammation. So I think, there is precedence for examining data, finding these signals, and the question becomes, what do we do about that information? And I think what we do about that information comes down to, is this something that's important for the community, for public health, and do we think there's any plausible mechanism? Paclitaxel does have this history in the coronaries. I've seen various, and the question is there enough of a systemic exposure, at least with the stents, perhaps, to cause some untoward effects? So. We don't have certainty yet, I don't think.
Yeah.
But, 1 second, Vishal, before you say, obviously in the coronaries it was endothelial dysfunction, right? We knew that was what delayed causing thrombosis, stent thrombosis, et cetera. So here, this is why I say that, because I don't see it, and you're right, it is probably prudent, and it is public health, and obviously it's a mortality signal. It's not anything else other than a mortality signal. But I just want to make a comment here before Guja goes up. So what, I don't know if the audience saw, what Dr. Guja did was he tracked the sheath into the aorta and then was able to deliver the stent through, which is very important technique in PAD, because now through all that plaque, you're able to get a sheath in and you're able to deliver it. We teach that in osteoiliac stenting as well as in renal stenting, and I think this is a good reason. So now we're going to go ahead and inject. I think you're going to have to inject from above and below in order to be able to see whether you've got the proximal edge of the lesion. You probably have to come back slightly, and the stent is way too much in the aorta, as you can see. So we're going to pull the stent back a little bit, because obviously if the lady ends up needing a bypass, you want to make sure that you're able to place the stent properly so that we can do interventions through the arm and the leg. She's fine, right? Okay. Little pain in the arm or the leg? So Usman, I mean, great thoughts. Vishal, any comments? I mean, I know I've been yapping away and taking up Usman's time.
No, it's good. That's why we have Usman up here to give us his perspective. But I think you're right. Usman is right. When it comes to mortality and defining the causality to it, we probably, and where the water is muddied right now, it'll be hard to do another meta-analysis and justify that there is no mortality difference. I think we'll be, that's my personal opinion again. We might be forced to do a randomized trial, like Usman said, power to for mortality per se, to really give us that definitive answer, which again, unfortunately, puts up 5 years back in line, because we probably need a 5-year data down the lane. So, well, I guess that's the part of medicine. You gain some, you lose some. So, we just have to find the right answer.
Okay, so
let's see what's happening with the subclavian now. But it's great. I mean, it's great to have discussions, talk, and get everybody's perspective to see from a clinical and a statistical perspective as well.
It's too far, right? It's too far. We're gonna, so you can see here, it's too far in, and we're gonna go ahead and pull this back a little bit into the sheet, and that's good right there. Okay, I'm gonna walk this back. Okay, that's good. So siento, mama. Okay, that's good.
I mean,
Ready?
just for the audience, like
Go up.
they said right now, you just define, but you should always define the vertebral and the lemma anatomy, which we did in the previous pictures, just to make sure we don't cover it. So
Liz.
right there.
So you can see we're going up with the covered stent. You have eccentric calcium, and don't think because you have a covered stent you're covered. Which is really a misnomer and a pun, for lack of a better word. So we're going to go up. No, you're fine. You're fine. No, but I think you'll just go ahead and you deploy to nominal.
Yeah.
What are you at now?
There's so much recoil.
What's nominal? What's nominal?
8.
No, I don't think so. 9.
It looks a little better, definitely.
9. 12. RVD. Go to 9. Go to 10. Okay. And I would go down. Now what we're going to do is, I think we've done a good job up top. So we're going to puff out now, make sure we have no perforation from the sheath, guys. Give me a little puff from the sheath. Why do I have no pressure in my aorta? Yeah, thank you. All right, so now I'm going to go forward with this.
So you just take the sheet.
Okay, now go forward with the balloon. Mm-hmm. You want to shoot the balloon?
Yeah, I would probably push the sheet over. Yeah.
We're already in.
Okay, good.
Pull
Now,
and pull,
okay, now
and
we're going to
then
go
now
ahead.
go.
Go ahead now. Go ahead with the balloon. Now, yep. Okay, good.
Now I'll go.
Perfect. Okay. Can you mag up, please, so I can see? I'm an old man. So bring it closer to me. Bring it closer to me. Cindy, please. Okay, the sheath is in the tip of the stent. Yep, that's good. Now we can get another balloon in. Yeah. Go
I
with
think we'll
another
just
balloon.
take a fresh balloon if it doesn't pass.
Yeah, I got positive. Yeah, well, I got it good. It's not going to slip. Okay, good, excellent. So at this stage, Usman, I want to give you a little update. FDA is going to meet with again with the Vascular Leaders Forum, and then what they're going to do is they're going to go ahead and discuss what are the next steps, and this is going to happen in mid-June. So at this stage, unfortunately, we are not using drug-coated therapy to go ahead and do any of these procedures. We are using it in high-risk patients who have had recurrent restenosis, and then we will go ahead and talk to the patients regarding this data, and just tell them that we've got this signal that we can't explain, and so that's the kind of situation where we're in at this stage.
Yeah, and I mean, and I think I think the FDA deliberately did not make a statement to not use these devices. And again, until you spend some time, particularly I think in these patients where you have no follow-up, we really got to figure out what happened to these patients, what's the vital status to get a proper assessment, and let's see where things land. And I think that's why they took that approach of being a little bit more cautious, convening a panel, and sending out that letter.
Yeah, I was surprised because they would have recalled the device per se. When you have increased mortality, the first thing to do is recall it and then look into it, which they didn't.
They did not, exactly.
So that is one thing, I guess, interesting that they took.
Right.
So why didn't they recall it, Usman?
Because I think of the uncertainty we're speaking about. I mean, you have a signal without a clear defined biological mechanism, and you have a signal in the presence of certain limitations. Minimal studies, low power, a lot of patients who were lost to follow up in 2 of the 3 studies for the 5-year data. This is enough enough limitations to not make any definitive statement. I think you can say there's a signal, and but we're not 100% sure of the magnitude and the size,
right?
And quite frankly, if it's even true or not. So until you sort that out, I don't think you can say, well, we have to stop using the devices. You can say we need to study it more, which I think is what they're doing.
So now you're post-dilating with a...
Yeah, with the 7, you can see the aortic plaque is so angry that we're really dealing with this. So now we're gonna go ahead and now track the sheath in, as the balloon comes out, so I can have access into the stent. And I think this is a very important, there you go, there goes the sheath. And I'm going to hold the sheath there as Karthik takes out the stent. And now what we're going to do is we're going to take a picture through the sheath. I'm going to hold the sheath there. No, I'm holding, believe me, I'm tight. No, it's not slipping, it's bowing, that's all. See?
See,
As you're
just
pulling it out. And also, I don't know where your wire is.
No, it's good. It looks much better. I guess the tension with the balloon is out.
So now what I'm going to do is, remember, you just want to work with the gradients. So I'm going to zero both. I'm going to go ahead, flush the aorta, please, and zero both, and then we'll see whether there's a gradient. So, and then once the gradients are done, we're going to just be done. So Yubi, I got to tell you, you've been phenomenal, and I thank you not only for spending the time here. I want to let the audience know that Dr. Baber is going to be at our FDA town hall. We call it Let's Talk Paclitaxel at Link Mount Sinai. He's kind enough to go over his thoughts on this meta-analysis and what we can do really to make sense of this data. And so honestly, I know Dr. Guja didn't say much. He was working really hard today, which he never does normally. That's another story. But I really appreciate you, Dr. Baber. Thank you very much.
Thanks for having me.
I know you're in the cat day, and I really appreciate you.
Thanks.
And Vishal, I think we had a lot of learning from this case. Just to kind of summarize, obviously to the audience, it's. In our part, it's our bad. We've been careful, looked at the subclavian, made sure that we didn't have a subclavian stenosis when we're planning a radial intervention, which I think is a huge point for all of us. I know the cardiologists also do that to make sure that there's no subclavian axillary disease prior to doing this. So I think that this was a big faux pas on our part. But we really clearly showed you how you can manage this. And now what we're going to do is 9 o'clock, but offline, what we're going to do is we're going to go down from the radial. We're going to fix the SFA so that this lady gets what she came for. But at the same time, she also got the subclavian fixed. Just 2 take-home points from the subclavian I think is very, very important. One is, in our opinion, in our humble opinion, I think that stenting the aorta ostial lesion from the arm is very difficult because obviously you have a huge pressure gradient, and any dye that you inject is going to flush up again, and you're going to miss the ostium. You can see by the pictures that we took here. Can you go C minus or fluorosave it or whatever you want? And then you can see by the pictures we took. You can see the aortic plaque is very aggressive. You can almost see the aortic plaque, what Dr. Guja was showing, and you can see that the plaque is almost is extending into the aorta quite a bit. Yeah. So it's very important for you. You can see the stent is tapered very clearly, and you can see how much black plaque there is. And I think that's part of it. At the end of every subclavian procedure, make sure you do an aortogram, that you haven't caused aortic dissection, and I think we'll end this case here. I want to show the pressure monitor and show everybody that the pressure is normal. I don't know why it's now decreasing. It was just even. We're gonna flush again. Let's zero again, guys. How do you zero like this? Zero both. Zero both. What's going on here? There you go. Zero both. There you go.
Zero both.
Let's zero both, please.
Yeah. Okay.
It's not zero, right?
Where?
There you go. So we have, let's see, 161 in the aorta, 163 in the aorta, and in the subclavian, I don't know what we're going to have here. I don't know why we're not seeing it. Can I see my subclavian pressure, guys? Phasic. Turn it off again.
You have to zero again.
You have to zero again?
Yes.
All right, let's zero again. Zero again.
Zero again like
Go.
this. Yeah. We zeroed.
Okay. Open. So we have what? 114, 162, 160.
Yeah, it's good.
145.
145, 150, 158. And she doesn't have a lima. So at this stage, I think we're done. This is going to really increase her feeling. We may further balloon that offline, and we'll be done with it. So again, I thank all of you. I thank Asma for doing such a great job in presenting the case. Sardar for his contributions to the case. And we'll go ahead and we'll see you guys next month we have another exciting case to show you. So I appreciate all of you to sign in, and thank you to the team.
All right, thank you, PK. Thank you,
PK.
everyone, and special thanks to Usman for
Thank
coming
you.
on. So for everyone, please log on to our website, ccclivecases.org. Under the peripheral section, you can see all our cases in the archive section on the tab. We'll keep you posted with further information on Link Mount Sinai, which is coming up in June 11th and 12th, main course, and a fellows course on the 10th to proceed. And we'll see you next month, April 24th, Wednesday morning, 8 o'clock Eastern Standard Time for another exciting case from Mount Sinai. Till then, enjoy your spring. Thank you.